Morning, folks. It's Tuesday, September 7th, 2021. This is the Podman community meeting. If you have not yet, please sign in with the HackMD. There's a link in the Blue Jeans chat for that. And I've pre-populated a number of people in there, but if your name is there, please go ahead and add yourselves. We've got a number of topics today to talk about. First off, we're going to be talking about the official Debian, official Debian Ubuntu packages updates from Lokesh. We're going to do a Podman machine update from Matt. And we'll be looking at containerized DNA analysis, if I could type correctly, inside the notes from Eric Bernoth. And then Chris Short, hopefully, will be giving us a video on using Podman in an IDE. I do have the link to the video in case it goes bad. I'm having issues trying to share it this morning, so hopefully I'll make that work. And then we'll have an open forum and topics and questions. We had planned to do a networking in rootless containers this morning, but unfortunately the speaker is having voice issues, so we've pushed that out to the October meeting, and we'll do that then. So apologies to those who came here looking for that one in particular. So I think that's all I have to say to start up with this, other than, one last note, I'll be taking notes during the meeting. If you see that I type anything wrong or you want to add to it, especially if you have links or slides that you'd like to share after the meeting, please add them to the notes. All right, so given that, I'm going to stop sharing and I will let Lokesh take over.
All right, can you hear me?
Yep.
Okay. So thank you, Tom, and hi, everyone. Since Reinhard couldn't make it to the meeting, I'll be reading out the Debian and Ubuntu updates he sent me. So first off, Debian. Debian has recently released version 11, aka Bullseye, which ships with kernel 5.10 and Podman 3.0. Since Podman is statically compiled, Podman 3.2 from experimental works fine as well, at least on Reinhard's laptop. The Debian 11 release means that Debian Unstable is now open for development, and upgrading all dependencies to support Podman 3.3 is well underway. If anyone is interested in helping out with Debian packaging, Reinhard would appreciate assistance on the following: identifying and upgrading package dependencies in Debian, filing bugs on what needs to be upgraded, and preparing package uploads on the GitLab instance at salsa.debian.org. That's about it for Debian. Now, Ubuntu. The upcoming 21.10 release currently ships with the packages from Debian Experimental, which means Podman 3.2. Since we're past August 19 feature freeze, Podman 3.2 is likely the version that Ubuntu 21.10 will ship with. So those were the updates from Reinhard. If there are any questions, I suggest you ask Reinhard directly. I'll add his info on HackMD. That's about it. Tom, back to you.
Tom, Lokesh is done if you can't. Now we can't hear you, Tom. Tom, we can't hear you. Yeah, Matt.
Okay, so
yep.
Sounds like Tom wants me to take over. So this ought to be a fairly quick one, but I just wanted to give an update on Podman Machine, where we stand, where we're going. So I am not really the right person to do this, but unfortunately the right people are both on PTO. So, oh well, I'll take it anyways. As you may or may not know, there has been some significant developments in the field of desktop containerization, let's just say, over the past few weeks. And because of that, we've gotten a massive influx of activity in the community around especially Podman Machine, but Podman in general. And I'd like to welcome all our new community members because of that. But anyways, Machine has been getting a lot of scrutiny, a lot of PRs, a lot of bug reports. We're fixing the bug reports as fast as we can. Just bear with us there. Most of the comments we've been getting are in regards to two different things. One of them is the ability to mount a Docker socket, a Docker compatible socket rather, natively onto the host, so we can use Compose on your Mac or Windows box and not have to worry about SSHing into the VM that we create for Podman Machine. That is a new feature request, but it's one that I think should be fairly simple, and it's one that I'd like to get out in a future release. More details on that soon. The second and more interesting one, in my opinion, is volume mounting. So, Podman Machine right now handles port forwarding into the VM automatically. It does not handle volume mounting into VMs automatically. This is something that we've done a lot of discussion around how we want to implement it, and I don't think we have a good answer yet. We're investigating various options for how to make the connection, SSHFS, VirtIOFS, as well as 9P and NFS. And we would like to get it to the point where Podman, where you do a -v when you create a container, it will automatically mount that directory into the VM, or vice versa. Regardless, it will get the content from the host into the VM automatically. How we do that, however, is still very much a topic for debate. So this is something that we are going to be discussing at a future Community Cabal meeting, I believe. Is that next week, Tom, that we have the next Community Cabal?
Yes, it is already, by a strange way it worked for the monthly lineup, but it's next Thursday.
All right, excellent. So we have a discussion topic for that at the next Community Cabal, where we're going to discuss the architecture that we'd like to go with and generally solicit comments on how to proceed with that. It could be that we merge something right now to get us some degree of support for this, and then we work on a proper solution in the future. But we will I think we'll try and come to a decision on that at the next Community Cabal. So if you are interested in volumes and Podman Machine, definitely make sure to attend that. I think we're pretty much set with Tom, so we can move on.
Yeah. And thanks for those who have opened up PRs for the support right now. It's obviously what happened last week was not something we planned. So we were hoping to work on this over time, but now the priority has sort of jumped quite a bit. So. Tom, you're muted according to the screen.
Oh, okay. This time it was my fault, not the machine, Blue Jean's fault. Eric, are you ready to go?
Yep.
Okay, all yours.
And again, there's a countdown here. All right. Can you see my screen? You should see a few of flies.
Yes.
So I have started a new project during my vacation. I met with some friends for dinner, and how weird things can happen. We talked about some interesting IT problems that they have. So what they are doing is, in a company here in Berlin, they are analyzing DNA. And the questions they try to answer is basically, what are these small markers? You can see this in the picture here. What are these small markers where a small changes to the DNA can have a big impact on the mutations of the individual? And here in this example, we see flies, like one picture in the middle, the eye color is different, and the wing shape can be different, and the size of legs can be different, etc. And all the differences that we have as humans are also identified this way. And this can be, for instance, used to analyze to which group of people you belong to. So like, like analyzing your ancestors, but not by reading the books and the birth certificates, but by analyzing the DNA. And yeah, so the DNA is a long string of different, I don't know the English word now, but different components. And there are 4 different ones. And after they're analyzed in a laboratory, you get basically a file. And then the analysis starts, and the analysis is a true IT topic that is quite interesting. It has a lot of similarity with search and strings, etc. And yeah, I want to give a short summary of what I learned about this for the moment. And maybe we can see some opportunities for containerization here. So first, I want to introduce some file types. The first one you can already see here. So I basically found on GitHub a tutorial. I will share the links later as well. A tutorial for one of the tools. And there are a few file types that are interesting. And this one basically collects a read of data, a read of DNA. And what is interesting about these is they don't have the whole picture. I go back to the other slide. So in this picture, here we see on the left side, there are these strings that are like looped around. And one of them has a lot of these data points basically. And in the lab, they can only make a picture of a small part of it. Like if we look on the picture, maybe they only do one from the 0.0 to the 13.0. And then they have another one from 48.5 to 54.5. And so you get all these points in this file format here. And this is called FastQ. Let me see if we have the name here somewhere. Here in the top right. FastQ. And it basically contains all the data points. And above the data points with the at, there's some metadata that is basically like the name and the place, not the place on the string, but the place in the analysis tooling for the biology analysis. And after the plus sign, there's a quality marker. So they have some ideas about how good the quality of the read is. It's never 100%. And we are not fully sure at this point where on the genome is actually this photo, where was it taken. So one of the interesting points that we want to do with that is we want to take a standard that is defined as standard genome. So it can also have mutations. It's not like the Adam, it's like some point was decided this is the standard for this kind of individual or this kind of animal. And of course, we have a few for humans as well. And then we try to analyze where does this picture that we have taken, where does it fit in? And this is stored in another format that we can see here at the end of this call. It's called SAMP. And there's a binary version for it too, which is called BAM. And we see an example of it here in the picture. And this basically takes very similar information. You can see at the beginning of this marked block 6 here, it looks very similar to the metadata in the FastQ file. And it basically takes these reads that we have in the other file, and it makes a guess used on some algorithms where this can be positioned. And of course, if you have a small snippet of a bigger picture, it can fit in multiple positions, right? And there's some guess, like maybe there was an error read in position 13 and it's still okay. Or it's a little bit like string search where you can have a misspelling or the word can be separated because someone was writing it incorrectly. There's a space in between, something like this can also happen here. So there's a guess and a likelihood that this will fit. There's a scoring. And this tool will basically make a list in such kind of SAM file. And then we go into these files. We use the BAM file then mostly because it's quicker. And let me scroll down a little bit to this point. And we can look at it, of course, also with software, but here in a visual way, we can look at it in an aligned way. So at the top, we have the position. Below that is a standard genome, basically, where we have all the elements already. And then below, we can see with dots when a read has matched this standard. And we see a different letter if there's something different. And the start of a read is basically when there's empty spaces changing to the dots, there's the read start. So in the beginning, the start of the reads moves a little bit to the left here in this picture. And a read ends when you have these free spaces, like on the right side, there's a small area where it's a little bit dark, right? And this is basically one read that was aligned by the algorithm to there. There's not a 100% guarantee it's there, but it's the best guess from the algorithm. and then you can look at this data and make a guess about do you have a mutation here or not. And of course, if you have multiple reads below each other, then your likelihood increases that you're successful. Like if we look in this picture here, we see after the 0.731, there is a T, and below there are a lot of Gs. So the standard genome basically has a T there, and the reads from a human, they all have the G. So this person has a mutation in this point. And these mutations then can be used to analyze basically where does this human fit in the different haplogroups of people that exist. And sometimes there are also very important markers that can decide something like your eye color, etc. And so there are two interesting parts basically that cost a lot of computing power and memory and storage, etc. And one is that you have these small pictures that you need to find basically in the big picture, where does it fit in? And since you have multiple, you also need to align them to each other. You can probably think about that this will be quite a big tree. And the second is that after the horizontal analysis, you have the vertical analysis that you look at very specific points and you look what kind of mutation do we have here? Do we have a mutation or not? And what kind do we have? And for that, we will have another file as well. And let me see if I can find it here. Probably not. So we look at it in Wikipedia. And this is a variant call format, VCF. Also some metadata at the top. And then we have a position and an ID. So if this is a special marker, it has an ID, it has a name. And if you have some experience in this area, you also know what some of these names mean. Then there's a standard reference, a standard value that is there, and the alternatives that were found in the read. Sometimes it's one option, sometimes multiple are found, and they all come also with quality markers. Okay, they come with quality markers because it's only a certain likelihood, right, that you found it or not. And so, yeah, these are basically the analysis, the transformations that we do from one file format to the other. And there are certain tools used. Let me find an example here. Here we have the script that is used in this company of my friend. And you can see, for instance, here in this area happens the transformation from the FASTQ file, so from the reads that come from the laboratory, the transformation to this aligned version of the different pictures. And this takes most of the time from what I've seen. And it does some multiprocessing already on one node, but of course it's always one computer. So if you have a very powerful computer, then you can do a lot of calculations. And if you don't have, like, one of the ones that we use is like a normal workstation computer, so a little bit better than a desktop computer. And it takes a long time until it's done, so multiple hours. And yeah, if you have a more complex problem, maybe even a day. And the second part is this vertical analysis. This is this pile up here in the end. And this vertical analysis is something, if I understood correctly, for one type of search that they are doing, it can only run on one processor at the moment. So there are some ideas. How can we basically structure this whole system that is not just running in a bash script, and when you add a new server or a new workstation or something to the system, that you need to manually install all the dependencies? Couldn't we containerize it and run basically in a more dynamic fashion this way? And yeah, so that's basically what I wanted to introduce to you. That's what I'm currently working on. And I'm interested if you have some similar ideas already, some similar projects, like compiling source code, for instance, is a similar topic, I think, or also big database, full text searches, something like this. Maybe we can think about it and discuss how we could containerize it. If I understand correctly, some of these tools here, they are compiled C tools. Some of the tools are available at least indirectly through Python libraries. So they come with these Coda or virtual end formats. If we have a standard for that already to containerize it, maybe we could look at that. And maybe some of them are already containerized and we just don't know about it yet. All right, that's what I want to give you a short intro about. And yeah, if someone is interested, let's talk about it.
You're bringing back memories, Eric. I worked on this like 15 years ago with PALP.
Awesome.
It was a proprietary system, and then I used the European molecular biological tools that were written in Perl, and they were just starting Python libraries at the time. And we did the exact same stuff, alignments, and then basically parsimonious trees to figure out which two things were the most related.
Yeah.
But
Awesome.
I see some use cases to like containerize this for sure, because like I mean I think you could probably set up standard like ways where people input output things, but I think it'd be a lot of magic with like bind mounts and things like that, so you get pass through data that's fast for when you're doing like the analysis and things like that. I don't quite know what you're looking to do though. Like, are you looking to like basically build like almost like a project on top of Podman that would be all these tools containerized?
So yeah, I look basically at as a use case. So as a Podman user, I look at it, not as a Podman developer. And I'm thinking about using Podman for this because we have a small amount of computer, and they are very differently structured. So it's not like in a typical OpenShift cluster where you have like 10 machines that are of the same type. Here we have 4 or 5 machines, and they're all very different. Like some have more disks, some have more CPU, and they have different kinds of CPUs. Like one is a workstation, the others are servers, etc. So it's a lot of individual environments basically. And currently we run all these scripts manually on them, and we need to install packages manually, etc. So it's also a topic like how can we get the packages all in a standardized fashion. And yeah, so I thought it's a smaller use case basically, so it would fit better with Podman than with OpenShift.
Yeah, that makes sense to me. I'd also love to be able to get the libraries in UBI so you could actually use UBI as the base image, but right now we don't really support C use cases. So like a lot of those devel libraries are all in rel only. They're not in UBI.
Yeah. So yeah, that would be also interesting. And as you also said with the volumes, speed also plays a role, right? If we have too many virtual volumes or too many abstraction layers between the actual storage, it might slow down the process so much that in the end we decide it might not be worth it. So maybe
I suspect the way those tools. Yeah, I suspect the way those tools do their analysis too, they probably drop. I don't remember. I mean, it's been years since I messed with this, but I suspect they're writing to the end of files and it would do all kinds of weird overlay stuff and things like that. So like, yeah, I would imagine bind mounts would make more sense, like pass through volume mounts.
Yeah. Yeah, that's it already from my side. Some questions or other ideas? Then I hand back to Tom.
All right, thank you, Eric. All right, so I'm crossing my fingers here, all my toes and everything else, and hope that I can get this video working from Chris Short. So let's give it a go here. Are you seeing the start of a video?
We're seeing you.
Oh, seeing me. Okay. Click to play. Oh, it says click to play to start sharing the video. All right, we'll give it a go.
Good morning, good afternoon, good evening. I am Chris Short, a host with the most of Red Hat live streaming. I'm here to kind of demonstrate something that I think a few people here might not realize, a capability that we have with Podman through VS Code, specifically VS Code's remote experience. So basically what that does from a system side is it reads your SSH config file. So me being on a Mac and the fireplace behind me having a Linux OS on it, I can essentially SSH into that box, create a VS Code environment to where I can do all my work from any machine to that box. So that box is actually my quote personal computer, and I'm on my work computer right now. So let's turn my camera off real quick and show you what's going on here. So what I have here is a VS Code extension, or VS Code with the extensions installed. If you're curious, the specific extension is right there, remote development. This can work on WSL, right? And it's a full pack of other extensions. So in theory, you can do containers with this. So I could set up a container with my dev environment in it all by itself. But for this instance, since I'm just SSHing into the box behind me, my personal computer where I'm doing work personally, or where I need a Linux instance for work stuff, for example, we do the point system uses a container to do all of its calculation for the level up hour. So if you've ever turned into the level up hour and we've talked about sweet speed internet points, you have seen the output of a Podman process. So just to let you know, right, like this extension's installed, you can see it. There it is, Remote Explorer. What this is reading is my local SSH config. And as you can see, I am a fan of Looney Tunes characters for naming, but also notice that I'm a Tailscale user, so all these instances have a VPN endpoint address as well, so I could be technically anywhere in the world and just remote into my local box and run everything that I need to run from my machine. I've already got this instance set up, but basically what it's doing is I'm SSHing into a box, VS Code spins up whatever it needs to spins up, and it just opens up this, port between the system behind me, the remote system, and it does all the SSH tunneling and everything else. So, I'll go through and I'll be like, oh, I need to add an extra, I need to add a points reference file in here, basically. Run, do a couple other things, administratively, and then I have this terminal instance that's on the remote box. So to prove that, like I'm on a Mac right now, you can tell just from the UI. Let's do this, OS release. Hey, look, that's a Fedora 34 box. So I'm fully in my IDE using that terminal on a Fedora 34 box, which means for level up points, if I go into the points directory, you see there is all right, so you see in the level up tools, get sheet directory, there's this make file. So I need to run a make podman build, and it's going to do all my podman stuff and give me all the, all the work that needs to happen inside that pod, pulling in other sheets from Google Cloud, et cetera, et cetera. Calculate all the points, and then spit out a latest sheet, which will be in the other directory. Well, not that directory. Which will be in the points directory here under current sheet. So just cat that out. Current points. .csv. And there's all the points for the people that have participated in the show. And all of this is done through VS Code on my Mac, just using that remote extensions client, or, extension to SSH into the system behind me. And all this stuff runs over SSH. Now, what I want to show you is something, like a typical use case, obviously, for, Podman. Let's just run a web server real quick. So I've already pulled down the images. Let's just tune up this web server real quick. Oh, it's already in use. Crap. All right, so since it's already started, it can easily show you the actual page from the remote machine behind me. And yes, you'll see all kinds of weird stuff because I have all kinds of weird stuff on my local network. But 8080, hey, it works. Taking
Chris,
this
dumb
a
question.
step further,
It's a video, Scott.
Oh, sorry, I didn't realize
It's
that.
a video. I'm sure he'd help otherwise. And you may see, I forgot to mention before we started off, some of the stuff is blurred out because there was information that he didn't want to give out, such as people's names and email addresses. So he's done
Okay,
stuff
yeah.
out in the video.
Yeah, I did wonder why it was blurry in certain places. Okay.
Yeah, that's what happened there. All right, I'm going to restart this.
close that real quick. If I wanted to edit my Hugo-driven website, so if you're not familiar with Hugo, it's a static website generator. I use it to run pretty much all my websites, including my newsletter, devopsish.com. Please subscribe, blah, blah, blah. But again, If I ever needed to, from my work machine, work on that remote box, I could be on Tailscale, sitting at a conference venue, and like, oh shoot, I was making changes. I had some stuff in flight. I need to get to that and work on it and commit it before the next newsletter goes out or something like that, right? Like I can totally do that from here. So I have multiple websites that use multiple versions of Hugo, but it's like one version versus the other. So if I do this actually spins up a local instance of Hugo running locally on the machine on port 1313, which is great, but it's on the other box and it's probably firewalled off. So give it a second to build here. It's trying to pull in tweets from half the internet, et cetera, et cetera. But look, VS Code gives me this wonderful little pop-up that's like, hey, we could forward your application port over here. Let me open that in the browser. Okay, let me just, come back over to localhost here and say, localhost, folks, I'm on my Mac, localhost 1313. There's my website. So there's some really cool capabilities in VS Code that I think we could utilize extensively with Podman. So thank you for your time today, and I look forward to seeing what other fun things people can come up with here. And this was designed to be a brain teaser, so hopefully you can get some ideas from it. Thanks.
Yeah. I think I've returned now. That was from Chris. He really wanted to be here to do it himself, to have questions and answers going, but he's got live video streams going on at the same time that this meeting goes on.
The audio sounded so good that I actually thought that was him, and what the heck? Like I was so confused.
Oh, I'm glad it came through well. I know it plays very well on my machine, so I was hoping it would as well. So Chris is very open for questions. If you want to ping him, I'm sure he can. Just give me a ping yourself if you don't have his email, and I'll hook you up. All right, so that's all I had.
I just wanted to interject that the Docker plugin, we have people using that against Podman through the RESTful API.
With VS Code, you mean the VS Code plugin?
Correct.
Yeah, yeah, that's cool.
It would be nice to get a libpod or Podman plugin to use the extended features of Podman at some point.
I had thought, Dan, that they had one that the Red Hat developer team was working on, but I don't. Okay. It's been so long since I looked at it, I don't know what's gone on with it.
Yeah, I've not played around with it much at all myself, and would like to. Got to find some time to do that because it looks really, really interesting.
It's not quite as old as my bioinformatics knowledge, but still pretty old.
Right. Do we have any other questions? Any topics? Or any other topics for that matter?
I have one, but I'm planning on. Last week I was on PTO, but I started fooling around with looking at Docker Bench, Docker Security Bench, and trying to convert that to Podman. So I'm planning on opening up a new container project, basically called Podman Security Bench, and taking a lot of the Docker Security Bench code and converting it. It's all written in Bash right now, and there's some interesting things, but it might be an interesting project to extend and eventually get shipping inside of Fedora and RHEL, because it was triggered last Thursday by there was a presentation at DevConf US where Ansible was firing up Docker Security Bench, and I said, geez, we should sort of plug into that world ourselves. And so I'm hoping once we get that up and running, people will look to help contribute to it.
Okay, sounds good. And for topics for next meeting, does anybody have anything? We obviously have the rootless container networking that we were going to do with Paul today. We'll